Hello students, I hope all of you are doing great students. Welcome back to your own channel Vedantu NEET English. This is Basavaraj Sir, your biology master students. Students, if I am properly audible to all of you and visible to all of you, quickly add any emoji in the chat right now students. If I am properly audible and visible to all of you students. If I am properly audible and visible to all of you. Now students, now let me ask you something students. Let me ask you something amazing. In today's class, I will be teaching you what is the science behind the curd making, what is the science behind dosa making, and what is the science behind making the entire cheese. What is the science behind the making of bread? So students, are you ready? See, there are two ways I can teach this chapter. I can make you by heart the lines. I can make sure you just mug up few lines and then be happy. Now, students, do you want to do that, or do you want to learn the interesting way, the amazing way, students? All of you, quickly, students, quickly in the chat. Do you want to know the actual scientific method, or do you want to mug up the lines of NCERT? Students, tell me. All of you, do you want to mug up the lines of NCERT, or do you want to learn the topic? All of you students. It's a very, very easy, very interesting topic, students. Very easy, very interesting topic. Okay. So students, shall we start, all of you? Shall we start? Yes? Okay. Perfect, students. Perfect. Now, first things first, students. First things first. Right. Do you think, students, microbes are harmful? All of you. Students, do you think the microbes are harmful to all of you? Yes, they're harmful. Obviously, microbes are harmful. Yes. But do you think microbes are useful to all of you? Obviously, students, obviously. So this entire chapter is based on the fact that microbes are useful to all of you students. Now, let me ask all of you in very, very simple question. Very simple question. When I say microbes, what is coming to your mind right now? All of you, just close your eyes. All of you, close your eyes. And I say microbes. What is coming to your mind right now, students? What is coming to your mind? Which organism is coming to your mind right now when I say microbes? 99% of time, isn't it always bacteria? Yes, sir, students, all of you tell me honestly. 99% of time, it is bacteria only. It is bacteria. Okay. Now let's take bacteria as an example of microbe. Now, can anyone tell me which kingdom do you think bacteria belongs to? Anyone, students? Anyone? Now, if you are thinking about your amazing bacteria belongs to which kingdom, students? Which kingdom? Anyone? Which kingdom bacteria belongs to? Okay. Now, I'll give you a small clue, students. I'll give you a small clue. Bacteria belongs to a kingdom where we find unicellular organisms and we find prokaryotes. Anyone, students? Anyone? Here we find prokaryotes, we find unicellular organisms. That is kingdom Monera, students. That is nothing but kingdom Monera. Okay. Now, there is one more famous example now, students, which has your fake legs. Not fake, yeah, fake legs. I was going to say fake personality, but that is different people, right? Fake legs, right? It has something called as pseudopodia. Students, which celebrity am I talking about? The celebrity which is in your syllabus from a very long time. That is your amoeba. So amoeba is also a microbe. So amoeba belongs to which kingdom, students? Amoeba belongs to which kingdom? Monera only? No. Amoeba belongs to kingdom Protista. There, Protista, we have unicellular eukaryotes. All of you know that. So we have kingdom Monera has microbes. Kingdom Protista has microbes. Virus, students, virus belongs to which kingdom? Students, anyone? Virus belongs to which kingdom? Can anyone tell me? This stupid thing called as virus, because of which you get cold, the rhinovirus. Which kingdom, students? Anyone? Think. Use your remaining brain cells and tell me, virus belongs to which kingdom? Sure, you'll be like, sir, no kingdom only. Virus, no kingdom. Radhe. Yes. Doesn't belong to any kingdom. So, but virus is also a microbe. Then prions is also a microbe. Viroids are microbes. There are microscopic plants. There are microscopic animals. So, students, when I say microbes, I am not talking about any particular kingdom. Yes, famous is Monera only. But when I say microbes, microbes are the organisms which are big or small, students. Which are big or small? All of you, big or small? Students, please remember, when I say microbes, I mean small. Students, I mean small organisms. I mean small organisms. Small. All of you, tell small. Okay, I mean small organisms. So these are the organisms which are very small. What do I mean by that? It means that we cannot see them with naked eyes. It means we need microscope to see them, students. We need microscope. We need microscope to see them. Yes, so students, very small. They're very small. So we need microscope to see them. That is called as your microbes. Now, students, tell me honestly. Okay, keep your hand on your heart and tell me. Okay, tell me, students. Do you think humans are greedy? Students, all of you, because I love this word called as small a lot. I don't know why small. Okay, so tell me, students, we humans are greedy. No, we are greedy. So we exploit plants, we exploit and animals. Students, we didn't even leave microbes also, right? Papa microbes, it is chilling in a corner. We didn't even leave microbes also, right? So we are so greedy that we are also using microbes for our benefit. That is what I'll teach you today, students. That is what I will teach you today, how microbes we are taking advantage of. Okay, that is a different topic. Okay, so how we'll take advantage of microbes today. Advantage of microbes, students. So microbes are your tiny living organisms that cannot be seen with the naked eye. We need microscope, students. We need microscope. Now, let me ask all of you one simple question, students, again. Do you think we have microbes in our body? All of you, tell me, students. Tell me. You cannot be like, sir, you cannot be like, no, sir, no microbes in my body. But do you have microbes in your body, students? All of you, tell me. We're from Mysore. Oh, nice, student. Mysore is an amazing city. No signals only. Everywhere, turn. Tell me, students, do you think we have microbes in our body? Students, there was a paper. There was a research paper, which I have a link also, students, because all of you deserve the best information in the world. There was a research paper. All of you can use the ID here, a PMed ID, and check also. This research paper told us that there is around, you'll be shocked, 0.2 kg of microbes in our body. 0.2 kg, that is around 200 grams of bacteria in our body only. So students, so if microbe is present in our body, do you think microbe is present on this pen surface right now? Like, is it possible that microbe is present on this pen surface? Yes, it is possible. Is microbes present in the water which you drink? If you do not have your nice filter in your house, there is a chance that microbe is present even on the water also. It is inside the water also. Yes, it is present in the water also. Now, I want all of you to look at this beautiful image. Students, I want all of you to look at this beautiful image. Very nice image, no? Very nice image. Very nice. Okay, very nice. Now, if you look at this image, students, do you think microbe is in the air, like in this air also? Do you think microbe is present in your water? Do you think microbe is present in the tree? Do you think microbe is present in the soil? All of you in the chat, tell me, do you find microbe in all the surroundings? So technically, what do I mean, right? Microbe is present everywhere. Zahir, microbe is present everywhere. I prefer messy always, right? It is present everywhere. Everywhere we have microbes. Okay. Let me ask you one more question. Do you have microbes in this hot, acidic environment, in this hot sulfuric acid, in this hot conditions, right? Acidic vents. Do you have microbes here? Students, I love messy. All of you know that, right? So, do we have microbes here? All of you. It is present. Microbe is present here also. Do you think we have microbes, students, in the ice? This is ice here. Yes. Do you think we have microbes here? Yes, we do. We have microbes here also. In the eyes also we have microbes. Yes. We have microbes almost everywhere. Okay, now students. Okay. So we have established microbe is small, microbe is present everywhere. That point is clear to all of you? Okay. Now let's do a small experiment. Okay. Now I want all of you to become one scientist now. Scientist. Now I will give you a microscope. Okay, don't break it, students. Don't break it. No. I will give you a microscope. Now in this microscope, in 10x, we might not be able to see any microbe. 10x is very difficult to see microbes. Now let's take 100x. In 100x also you might not be able to see something. In 1000x, yes, we can see, students. From 1000x magnification, we can see some bacteria. We can see some bacteria here. Bacteria seen. Okay. Now if you go 1000x magnification again, we can see a little bit of fungus. Then we can see protozoans, that is your amoeba. Then we can see viruses in your higher magnification. We can see prions, we can see adenovirus, we can see bacteriophage, we can see tobacco mosaic virus. So students, next we can see viroids, prions. So next time when someone says microbes, you need to start thinking any organism whose size is less, very tiny. We have bacteria, we have fungus, we have prions, protozoans, viroids. All of these are, students, all of these are microbes. All of these are microbes, students. All of these are microbes. If you look at your NCERT, it says here, students, in NCERT right now, they have given you certain diagrams here. This is your red color. Technically here, there is gram-negative bacteria, gram-positive bacteria, students. Here we can see this gram-negative bacteria is a rod shape. It is rod shape. Students, can anyone tell me in the chat right now, what is the other name for rod-shaped bacteria? Anyone? Science world? Yes. Then we have Ravi, Esther, Sony, Asit, Ravi, Lakshmi. Tell me, all of you. ZXLR. Tell me, what is the other name of rod-shaped bacteria, students? Rod-shaped bacteria is also called as bacilli or bacilli shape. It is also called as not cocci. It is called as bacilli. This spherical shape is called as cocci. So this is cocci, this is bacilli or bacillus. Okay. Now students, here we can see a rod shaped bacteria. Here we can see flagella coming out from here. Students, if a bacteria has a flagella, do you think it is motile or non-motile? Obviously, if it has bacteria, it is going to be motile only. It is going to be motile, students. Here is something called as bacteria. Students, we are scared of bacteria. Bacteria is scared of bacteriophage, because this bacteriophage can infect even bacteria only. It is the ultimate big boss here. It can even infect the bacteria itself. Okay, here we have your TMV, tobacco mosaic virus. Yes, rod-shaped bacteria, students. Here gram-negative, I have given you a different image. Gram-positive. Gram-positive is always violet color because of the crystal violet. Saffron because of the pinkish red color here. Now students, how many of your pets in your house? Raise your hands. How many of your pets in your house? Pets. See, I have a dog in my house. Okay, I love him a lot. His name is Cash. I have a dog named Cash. Okay. Now this boy in my house, right? I got him a dog bed. Okay, I got him a dog bed. This person, my Cashew, he doesn't want to sleep in dog bed. He's still sleep in my bed only. He'll still sleep with me only. Now I understood by that, every single organism, it could be a dog, cat, everything, it has a habitat. And I made you realize that bacteria is present everywhere. Bacteria is present, students, everywhere it is present, bacteria. Okay. But is it possible to grow bacteria in a lab, students? Do you think we can grow bacteria in a lab in a controlled environment? Yes, it is possible. Yes, it is possible, students. We can grow bacteria on a medium. So we can grow this bacteria in a petri dish in a medium called as nutritive media. We add water, agar, salt, sugar, vitamins, amino acids. When we add all of these different components, we can grow it in a Media here. Grow it in a media right now, students. In a media. Now, students, can anyone tell me where do we get this agar agar from? From where do we get? Students, where do we get agar agar from? Anyone who can tell me? Agar agar is obtained from some type of your red algae, no? Your Phaeophyceae algae, Gracilaria and Gelidium. Very good. From Gracilaria and Gelidium, we get this agar agar. Okay, heteropolysaccharide. Okay, students. Now, students, all of these microbes, right? All of these microbes, when we grow it on a media, right? We can see them as larger colonies. These are called as bacterial colonies. So, if we grow them on a media, we can see large colonies everywhere. And these colonies, students, these colonies can be seen with the help of naked eye. For this, we do not need any microscope now. See, to see these, we need microscope. But when they form a colony, when they group up, we can see them very clearly, students, very clearly. Now, students, with that being said, now, shall we start the advantages, how we have taken advantage of bacteria, students? Shall we start? All of you. Now, I will teach you the scientific way, students. Please focus very well now. Now, I will teach you the scientific methods. Ready, all of you? Ready, students? All of you ready? Now, I will teach you the scientific ways of learning this. How you can make curd, how you can make bread, scientific way. Okay, students? Yes, we can make from potato starch also, yes. Yeah. Now, students. Now, first thing is called as your curd making, students. The first thing is called as curd making. So, please understand, very interesting it is, very interesting. I can just make you read, students. What I can do is, I can make you read the lines of NCERT. I can be like, see students, all of you know, curd can be made by LAB. Here I'll read a line, see students, there's a line here. It says, during the growth, LAB produces acids that coagulate and partially digest the milk, protein. Now students, reading is very easy. But to make you understand the process, I will get satisfaction there. So students, shall we start? All of you. Students, shall we start the entire process of curd making? Let's start. Okay, students. Now I want all of you to understand. To make curd, we need a main ingredient. The main ingredient to make curd is what, students? Curd is made from what? All of you know, you make curd in the house, no? Or you buy packets. To make the curd, we need the milk in general. So milk is our main thing here. To this milk, we add, students, this is the milk right now. To this milk, we add a spoon of curd. To this milk, we add a spoon of curd now. Now, students, I want all of you to understand what is present in the curd, what is present in the milk first. Okay, please understand. If you magnify what is present in the milk, a milk has the proteins in it. All of you know, milk has protein. It has casein protein. It has whey protein. It has casein protein, whey protein. It has fat globules, which we can see on the top usually. It also has something called as lactose sugar, which is a disaccharide here. It also has certain ions. So all of this is present in the milk. So please remember, there is protein, there is fat globules, there is lactose sugar, mainly. Remember this much. Okay. Now this is present inside the milk right now. Now students, what is present in the curd now? All of you know what is present in milk right now. But what is present in the curd now? If you look inside the curd, there is a very famous bacteria called as LAB or LAB or lactobacillus bacteria. This is your, students. This is called as LAB or lactobacillus bacteria. Okay. Now students, what is fat globules? Fat globules are basically lipid molecules. Lipid molecules are present. Okay. Now students, lactobacillus bacteria or LAB is present where? In the curd right now. In the curd. Now, what will happen when we take the curd and mix it into the milk? Okay, students. Now, I want all of you to remember here, the curd is also called as, students. The curd is also called as inoculum or starter culture. Starter culture. Inoculum or starter culture, students? Inoculum. Or starter culture. That is your curd. Okay? Now, students. Now we take this curd and we add into the milk. Now this lactose was present in the milk. Lactobacillus was present in the curd. Now, please understand this. One second, students. Please understand, students. This entire lactobacillus will attack the curd. So this entire lactobacillus will attack the lactose. Now, lactose being a disaccharide is made up of two monomeric units. This disaccharide will break. This lactobacillus, it has an enzyme called as lactase enzyme. It has an enzyme called as your lactase enzyme. Lactase enzyme. This lactase enzyme will break the lactose. It will completely break the lactose. See this, students. So lactose is broken by lactase into glucose and galactose. Now this glucose again will get converted to galactose, will get converted to glucose eventually, and this will now undergo a process called as glycolysis. After glycolysis, at the end of glycolysis, we get pyruvate. What do we get? We get pyruvate. So the pyruvate from lactose, we have now pyruvate. Now this pyruvate under anaerobic condition. What is, students, anaerobic condition? Anaerobic condition is lack of oxygen. Whenever there is a lack of oxygen, this pyruvate is eventually converted to lactic acid. So students, from your lactose, we have lactic acid now. Lactose got broken into pieces into glucose and galactose, which underwent glycolysis. Then we got pyruvate. This pyruvate is converted to, that is your lactic acid fermentation, lactic acid. Students, any doubt till now, students? Any doubt till now? Ask me quickly, students. Any doubt till now? Students, any doubt till now? From lactose, we have lactic acid, students. From lactose, we have lactic acid. Any doubt here? Any doubt here? Okay. Now, students, it is an acid. This is a acid here. This acid will drop the pH in the milk. In the milk, entire pH is dropping. Complete pH drop in the milk. Now, all of you know, I reminded you that milk has casein protein and whey protein. This casein protein and whey protein, proteins get denatured in low pH. Now, if proteins are getting denatured in low pH because of increase in lactic acid, the protein will start to coagulate. They'll make a groups here. When protein undergoes coagulation, because of increase in lactic acid, drop in pH, we end up getting the curd. Students, how is the process? I'll repeat again, students. I'll repeat again. First, we take milk. Inside milk, we have whey protein, casein protein, lactose, fat globules. We add curd to it. Curd is called as inoculum or starter culture. Inside the curd, we have lactobacillus. This lactobacillus will attack the lactose, break it in because of the enzyme lactase. So lactose is broken down to glucose and galactose. Glucose and galactose will undergo glycolysis to produce pyruvate. Pyruvate in anaerobic condition will become lactic acid. Because of the increase in lactic acid, what happens? The pH drops. If the students, if there is a decrease in pH, right? pH is dropping to, let's say, 2 or 3, because it doesn't go that much. If it is dropping, let's say, to 6, if there is a drop in pH, what happens? If there is a drop in pH, the proteins undergo denaturation. That is, they become simpler. Can you see? Denaturation. When proteins undergo denaturation and they end up coagulating, they make lumps, tiny lumps, right? That will lead to the curd formation, students, curd formation. Now, students, do you have certain friends who are lactose intolerant, who do drama all the time? They're like, no, bro, I can't drink milk. No, bro, this has milk. I can't drink milk. But students, technically speaking, students, technically speaking, right? If someone, unless they are very severely allergic to lactose, this curd right now is technically lactose free. Yes or no, students? This curd right now is lactose free. Why, students, it is lactose free? It is lactose free because all the lactose is broken down to make this curd. Students, understand, the curd generally doesn't have lactose. Lactose is present where? It is a sugar. Lactose is a sugar present in the milk. And that is broken down by LAB. So we end up getting curd. And this curd has amazing properties. Curd has amazing properties. Increase nutrition value of vitamin B12, easy to digest, probiotic for gut health, because safe to consume in lactose intolerant people, enhanced texture and flavor. Very tasty, very tasty it is. Okay, so curd is safe. It is lactose free generally. Okay, lactose free generally, lactose free generally. Okay, now students, now. How many of you love dosa and idli, students? It is grouping because all the proteins are denatured now and they end up combining because of low pH. Okay, now students, how many of you love dosa and idli? Now I will teach you how dosa and idli is made. Now the first question here is now dosa and idli undergoes what? Fermentation. But have you ever noticed your mom adding any bacteria into the batter? Students, all of you know, if you want to make idli and dosa, right? If you want to make idli and dosa, right? You need to do fermentation. But have you ever observed your mom adding any bacteria into the mixture? Students, tell me. Have you ever observed? Students, please smash the like button, all of you. All of you students, smash the like button. Sir, what is probiotic? Probiotic drinks. Probiotics are pro means good for your biotic, good for your health. Right? Now students, for gut health basically. Now, have you ever noticed your mom adding any type of your bacteria inside the curd to make the dosa batter? No, right? Then I'll teach students. I'll teach you what happens. You can tell to your mom also later on. Okay? Now students, have you noticed? Let's say Monday your mother is making dosa. Does she prepare the batter one day before? So have you noticed that? If you have to make dosa on Monday, right? One day before you have to make night. Your mother, if you're watching TV, let's say Big Boss is playing. Big Boss you're watching on Sunday night. Suddenly from the kitchen one sound will come. Kitchen sound will come, right? Have you noticed that? The sound will come only one day before the dosa night. Why? Because in the night time, magic happens. Okay, students, let's say this is a blender here or a mixie. Here I will add rice into it. I will add black gram into it, urad dal. All of you know urad dal, right? Urad dal, right? I add water into it, right? But I am not adding any bacteria because bacteria is actually present in this urad dal. Bacteria is present in this urad dal itself. Urad dal also itself has a bacteria. Now students, once you make the batter, idli dosa batter. Now idli dosa batter has carbohydrates. From where? It has carbohydrates from the rice. From the rice it has the carbohydrates and it has bacteria now. So this bacteria will eat the carbohydrates and eventually undergoes fermentation and batter becomes bigger overnight. It becomes thicker overnight. And next day you can enjoy this batter. Now students, have you noticed one thing here? When do you think your mother adds salt into the dosa batter? You go and ask students. You go and ask today. You go ask your mother right now also if you want. She will never add salt in the night time. Salt is always added on the next day morning for the dosa batter. You know why? Because if you add the salt one day before, the salt will change the osmotic concentration of the dosa batter and the bacteria will not be able to grow. To make sure the bacteria grows very well, we add salt in next day morning. Next day morning we add. Obviously, students, puffiness because of CO2, because of fermentation. Lactic acid fermentation is happening. No, fermentation is happening. Yes. So we add salt next day morning because we do not want bacteria growth to be stopped. Okay, students, did you ever think so much of science is behind dosa making? Students, do you ever think like this much of science for making one dosa? Students, the same concept also applies to the flavored yogurt, dokla, pickles, cheese, buttermilk, lassi, dosa, and everything. Everything has a bacteria called as your lactic acid bacteria. Okay, now yogurt and all, we have multiple steps in yogurt, but that is a different story. Okay, students, clear all of you? How to make dosa, how to make curd? Done, students? Done, all of you? Done? Yes or no? All of you? Done? Okay. Now, students, I'll teach you now how to make Tom and Jerry cheese. Ready, all of you? Students, I can make you read these lines very easily. It is very easy to read lines. All you need to know is to make curd we need LAB. To make dosa batter we need LAB. But where is fun in that? We learn science because we learn biology because we love science, right? All of us love biology, and to know what actually happens in a process, it's so much fun. It's so much of fun to know how things happen, right? Now this is your Tom and Jerry cheese, yes or no? Now students, here what happens, you understand here in Tom and Jerry cheese. Here also cheese is made from where? Cheese is made from milk. Milk has lactose. This lactose is broken by the bacteria called as your lactobacillus bacteria, LAB. When it is broken down by LAB, we get lactic acid eventually. Students, same process, we get lactic acid eventually. Now, in the case of Swiss cheese, this lactic acid is actually used by a bacteria again. It is used by a bacteria called as Propionibacterium shermani. Students, Propionibacterium shermani. This bacteria will break the lactic acid into propionic acid and carbon dioxide. And this carbon dioxide is stored in this cheese. And when the cheese is aging, as the cheese becomes older and older, this CO2 which is trapped in these holes, it will escape out. When the CO2 escapes out, whatever the region where the CO2 is kept, no, that will make the small holes in the Swiss cheese. Crazy, no? So this actually small holes is because of escaping of the CO2. See, this much all of you know, but now you know this also. How is it happening? So from lactose, from the milk, LAB, lactic acid, Propionibacterium shermani will act on the lactic acid, give propionic acid and your carbon dioxide. This carbon dioxide is trapped here. As the cheese is aging, this CO2 is escaping. Students, look at the science behind all this. Crazy Swiss cheese. Now students, now we will start with how can we use fungi for household products, students. Shall we start? Oh students, shall we start now? How you can use fungus now? Students, shall we start? The fungus which we'll be using right now is called as baker's yeast. It is called as baker's yeast. Ready, all of you? Students, ready? Students, I love science. That's why I teach in detail. You might have noticed my every class is very detailed class I do. Because I don't want to just come and teach you lines of NCERT and make you mug up something and go. My job here as a teacher is to make sure you love the subject also. Okay, that is what I'm doing. I want all of you to love this subject. I know this chapter is very easy, but there is always fun in learning something. Now students, now how to make bread. Ready students, ready? You can try it at home also tomorrow. How to make bread. Okay, now to make bread we need flour, atta we say in Hindi, right? Flour. Let's say wheat flour, we add wheat flour. We add maida or wheat flour we add. Maida also is wheat flour only refined, that's all. We add flour. Then we add sugar, we add salt, we add oil, we add water. Yes or no, students? Yes. Now, once you add water, right, and you do something called kneading, you make one nice kung fu moves. You do nice kung fu moves, and you make one ball there. Okay. Now, once you make this ball, what happens? Now, inside the wheat flour, actually, inside the wheat flour, there is a molecule called as gluten. Students, you might have heard about this. You might have heard, I want gluten free. I don't want gluten in my food, because many people cannot digest this gluten in their body. Okay, a little bit fancy people, okay. So this gluten molecule is there. When you do the kneading, right, when you do the kneading, this gluten molecule will make a mesh. This gluten molecule will make a mesh like this mesh, right? This gluten molecule will make a mesh, and in this mesh, what happens, students? CO2 is trapped here, papa. This CO2 is trapped everywhere inside this mesh. CO2 is trapped everywhere. Okay. So gluten is a molecule. Gluten is a molecule. It is a type of molecule, gluten. It is found in your wheat flour and everything. You get gluten in chapati also. Chapati has gluten in it. Okay. That's what answer. Now CO2 is escaped here. Now once you bake the bread in oven, you bake the bread. Now what happens? Wherever we had CO2 inside the mesh, all this CO2 will escape. All the CO2 will escape when you do baking. So the students, that's why, have you noticed bread is like a mesh? Students, have you noticed when you buy bread from your market, it has small holes everywhere? Students, have you noticed that? Students, how many of you noticed? Raise your hands. Students, how many of you noticed a mesh in your bread? Have you noticed? Bread has small holes everywhere. It has a mesh. That mesh is because of gluten, and these small holes on the bread is because of CO2 was trapped there. CO2 was trapped in small holes everywhere. So CO2 was trapped there. So that is how you make bread. That is the science behind bread, actually. The science behind the bread. Because of the gluten molecule, we have the mesh, and we have small holes everywhere because of the CO2 escaping. Students, crazy, no? Yes, no, students? Then, students, all of you understood? Yes. Yes, students, last we'll go to this class, students. I know. Students, what is happening? This in the bread we add yeast, right? Yeast is sugar is broken with the help of yeast. We get ethanol and we get CO2. CO2 is being trapped. Same thing, students, same thing. Now, your Prashant, right? To make this Prashant, to make this donuts, to make this bread, all of this has same, students, all of this has same process only, same process. To make this croissant, that is Prashant here, right? To make this bread and donut, we use the same technology, right? Same technology, friends. Now, next thing is blue cheese. Students, ready? Students, I can eat bread, I can eat curd, I can eat dosa, but God promise, I cannot eat this blue cheese. Students, this blue cheese has fungus in it, like penicillium fungus. Yeast is still fine. It has blue fungus everywhere. It has a little bit of like small insects also, some maggots and all it has, like a blue cheese. So this blue cheese is called as Roquefort cheese. It is called as Roquefort cheese because of the penicillium roquefort, right? Roquefort is a fungus which is present in this cheese here. Okay, cheese has fungus in it. This, see, yeast is still fine. You can't see it much, but this has fungus in it. Like proper fungus it has. Penicillium roquefort. Okay, students. Okay, students. Now, there's a process to make it, students. We use milk. We add rennet to coagulate everything. Curd formation. Then we'll drain the liquid out. Then we add this thing here, the entire penicillium. Cheese allowed to ripen for many days, and then we get blue cheese. So blue cheese is different from Swiss cheese. Swiss cheese is nice. Swiss cheese is very tasty, but blue cheese, very weird. It is very weird. Now, students, now we come to beverages, right? The Shinchan's father, students, have you seen? Shinchan father will be drinking some juice, no? Juice. It is your yellow juice. Okay. Now, how to make this yellow juice? Okay, students. Okay. How to make the yellow juice? Okay. How to make this yellow juice? Okay, students. Now, It is generally made with the help of something called as in the your native areas, in the old villages they make, in villages they make. There's something called as sugar palm trees. Now in the sugar palm trees, what happens? One uncle will climb it, nicely he'll climb it, and once he climbs it, he will add a pot. He will take a pot, he'll keep the pot here, and in this pot he will make sure he'll make a cut here. He'll make a cut here. From this date palm, there will be a liquid coming out that is called as sap. This sap is added into this pot here, and he will leave it there, and he will just leave it there. Now because of the microbes present in the environment, this sap of this date palm undergoes fermentation, and later on toddy is served fresh. Ta-da! That's how you make toddy, students. That's how you make toddy. Students, here is a question. I want who can solve this question. That's how you make toddy. That's how you make toddy, students. Toddy. Bread lover, huh? Nice, students. So I'm a cheese lover. Let me show you what is the answer. Conversion of milk into curd improves its nutrition value by increasing the amount of vitamin B12. Yes or no, students? Vitamin B12. Okay. Now, students. Now, right, toddy is an alcohol. Toddy is a type of alcohol, like whiskey, brandy, like that. It's a type of alcohol. Toddy is also alcohol, but it's called Hindi. We say desi daru. They say desi daru. In Hindi they say, right? In Kannada, students, what do we say in Kannada? I forgot, students, in my Kannada only. Students, what do you say in Tamil and Telugu also? All of you. What do you say toddy in Tamil and Telugu? You might have seen in movies. In movies, have you seen? Right, movies that one white color bottle will be holding white color bottle. White color bottle is old uncles in the villages. White color bottle, that is toddy. Okay, that is called as toddy itself. That white color bottle they'll be holding. No, they'll be hitting everyone with that water bottle when fighting will be going on. Right? Yes or no, students? Now, students. Now, should we start microbes in you? This is the last topic today. Last topic, students. I will teach you this entire thing and we'll stop. In the kallu, oh, nice, students, nice. Right. Now these three in today's class. Next class I'll teach you STP. Okay. Next class is going to be very important. In the next class, that is day after tomorrow. Day after tomorrow, I'll be teaching you STP. Sewage treatment plant. I'll teach you day after tomorrow. Okay. Kallu in Malayalam also. Okay. Okay. Okay, students. Students, ready for today's now last stretch? All of you. Students, ready for last stretch? Shall we start? Students, let's start. Last stretch for today's class. After this, it's over. Very few information is left. I'll teach you this few information, and you're good to go after that. Students, shall we start? All of you. Students, shall we start? All of you tell me yes or no, all of you in the chat. Students, first of all of you smash your like button. First of all, students, all of you smash your like button. This is my third class from today. Third or fourth class from today. I had a class from 3 to 4:30, 4:30 to 6, 6 to 7:30, then 8 o'clock I've started a class. Okay. This is my fourth class of the day. So I hope all of you are energetic because I am trying to bring a lot of energy into the class. So all of you quickly smash that like button right now. And I want to see all of your comments. All of you become very lazy nowadays. All of you don't comment also. Okay. Students, I don't have pani here. There's no water here in my room. I have to run and get water from somewhere. No water also. Because water has microbes. Now, students. Now, what about fermented drinks? Now, what is the bacteria involved? Students, what is the bacteria involved in fermented drinks now? Now, for an example, all of you know baker's yeast. This baker's yeast has a cousin brother. Students, this baker's yeast has a cousin brother also. All of you. This baker's yeast also has a cousin brother. The cousin brother is called as your brewer's yeast. Okay. Brewer's is the cousin brother here. Saccharomyces cerevisiae. Okay. Saccharomyces cerevisiae is the yeast thing only. Okay. Cousin brother here, students. Now, because of this cousin brother, with the help of this cousin brother, we can make all of this adult juice. Okay. Adult juice, this is adult juice. Beer, wine, brandy, rum, whiskey, all that can be made with the help of brewer's yeast. Okay, students. Now, how do we make it? Okay, how do we make alcohol? Students, do you want to know, or it is too much for all of you? Students, do you want to know or not how to make alcohol today? All of you. Students, quickly smash your like button, I'll teach you. Students, quickly smash your like button, I'll teach you how to make alcohol also today. Okay. Students, shall we start? Students, shall we start? Yes. Now, generally in your industries, there are breweries. These breweries have something called as fermenter. Fermenter is basically used to make large scale alcohol. Large scale alcohol is used to make fermenter, students, fermenter. Okay. Now there is something called as alcoholic fermentation. Now what actually happens in the case of alcoholic fermentation? Understand. Okay. In alcoholic fermentation, first thing we need is a source of carbohydrate. Source of carbohydrate we need. Okay. Like let's say in grapes is used in wine. We use barley in the case of beer, right? That is your carbohydrate source. Let's say one type of carbohydrate is your sugar, right? Sugar is a carbohydrate. Sucrose, right? Or let's take glucose only, students. From this sucrose we'll make glucose. This glucose now undergoes something called as glycolysis. Students, can anyone tell me how many steps are there in glycolysis? How many steps? Plant physiology, tell me. 10 steps. Very slow you are. Now, after this glucose undergoes glycolysis, we get pyruvate. Now, this pyruvate undergoes alcoholic fermentation to produce ethanol plus CO2. That is what, student. That is what is happening, right? Next class is the day after tomorrow. Next class is day after tomorrow, mine. Day after tomorrow. We'll finish the chapter. No, day after tomorrow, we'll finish the chapter. Day after tomorrow. Okay. Today is Wednesday. Friday is my class. Friday and Saturday, both the days. Okay. Now, this carbohydrate is getting converted to ethanol plus CO2. Ethanol plus CO2. Right. Now, this is what is happening, student. This is what is happening. In this fermenter, we add our entire structure. Here we can see CO2 is being released here, and we get the alcohol. Now, students, I want all of you to understand. Naturally speaking, the alcohol percentage will never exceed 30%. So, 13%. 13% is natural, students. Okay. Sir, Gopikam is laughing on you. Ma'am will always laugh. Now, students, natural speaking, alcohol cannot be made. Naturally speaking, alcohol can be made 13% naturally. 13% naturally it can be made. Why? Because if it goes more than 13%, the yeast will die. That is called as yeast poisoning, because they will poison themselves. See, alcohol is poison. Yes or no? Alcohol is poison, right? It is poison. So the yeast will poison itself and it will die. So naturally speaking, the maximum concentration of alcohol can be 13%. Okay. But students, have you noticed in the market there is whiskey, brandy and everything? We have 10% alcohol, we have 30% alcohol, 40% vodka, 50%, 70%. Students, I do not know who is drinking 70% alcohol now, because in esophagus one hole will become whole. In the stomach one hole will become in stomach also. 70% alcohol if you drink. Okay. Now, how do we make this concentrated alcohol then? See, naturally we get 13%. If it goes more than 13% alcohol, yeast will die. Then how do we make this concentrated alcohol? What is the secret behind it? Students, anyone knows? Students, anyone knows how to make concentrated alcohol? Anyone? Don't try at home. That is called as, anyone? One second, student. Let me just, my phone battery is low. Anyone? I'll give you time, students. I'll give you time to think. Tell me, students. No one knows, sir. Okay, I will tell you. Because of fractional distillation, students. Because of fractional distillation. Understand what happens. I have to teach. I'll teach you everything, students. Don't worry. Let's say here I have I'll keep a container here. Okay. Now I will make a tube here. This is a basic setup. Okay. Now let's say here I have alcohol here, and in this alcohol there will be water also in it. That is why alcohol percentage is only 13% with this water here, 13%. Okay. Now students, we will try to use the ability called as difference in boiling point. Students, what is the boiling point of water? Tell me students, what is the boiling point of water? All of you, 100 degrees, yes or no? 100 degrees centigrade. Okay. Boiling point of alcohol, let's say, is around 78 degrees centigrade. Okay. Now let's say I will put fire here. Fire. There is fire here. Now tell me students, which liquid will boil faster? Tell me students, which liquid will boil faster? Anyone? Do you think alcohol will become into vapors or water will become into vapor very fast? Tell me students, since alcohol has lower boiling point, don't you think so? Alcohol present here will evaporate first, and we will make sure the temperature does not increase more than 90 or 95, because we do not want water to be evaporated. So all the alcohol will evaporate nicely, and here we will cool down. So here we get what? We get here concentrated alcohol. Yes or no? Here we get concentrated alcohol here, because entire alcohol is evaporating, and here what do we, students? Remaining part here is only now water is remaining here. Water is remaining. All the alcohol is this side. Students, crazy, Okay, don't try it at home. Okay, don't try it at home, students. Don't try it at home. Okay. Now, students, how do we have so many varieties of alcohol? What is the reason behind varieties of alcohol, students? The main reason for variety of alcohol is the different types of ingredients used. Let's say wheat is used in the beer. We use barley in the case of whiskey. Rum is generally made from sugarcane. Grapes, wine. Vodka, potato. Potato is vodka. So every single alcohol has a different types of main ingredient, that is carbohydrate source. Depending upon the carbohydrate source, there are multiple varieties also. There are multiple varieties, students. Okay. So we have non-distillated, students. Anyone examples? Examples of non-distillated is beer and wine. And then we have distillated, where we have your brandy, whiskey, as well as your rum, and even vodka also. They're distillated. Okay, they're distillated ones. Okay, students. Now, next we come to, this is done. Topic is done. Now we come to antibiotics, students. Students, has any one of you ever taken antibiotics in your life? Raise your hands. Because today I will teach you how antibiotics was discovered, and I will also teach you types of antibiotics. Students, ready, all of you? Ready? Students, ready? All of you ready? Types of antibiotics. All of you. Students, quickly smash the like button first of all. The target is 50 likes. All of you, quickly smash the like button right now. Paneer Selvam, paneer. Okay. Rice can also be used, obviously. Rice can also be used. Rice has carbohydrates, right? Rice can also be used. Now, what is the meaning of antibiotic, students? Anti means against. Anti means against. Biotic means life. So antibiotics are basically against life, but they're against life not for us, for bacteria. Okay, so antibiotics are against life for bacteria, students, bacteria. Now, if I have to give a definition, students, write down the definition. Okay, so antibiotics are basically the chemicals. They're basically chemical substances. They're chemical substances which are produced by the microbes that kill or retard the growth of other microbes. Just word stop. Stop the growth of other microbes. Okay, that is student. That is basically the antibiotic student. That is basically the antibiotics. That is basically the antibiotic students. Now let's understand discovery of antibiotics. Now discovery of antibiotics was actually by laziness. Did you know that? Did you know that antibiotics were discovered by laziness, or let's say someone forgot about them? Now what actually happened was, all of you know that the scientist called as Alexander Fleming. Alexander Fleming was doing an experiment on staphylococci, right? He left the plate petri dish in the sink without washing them. Laziness, not but laziness. Now after what happened? After some time he observed that, wait a minute, why is bacteria not growing only? He observed that on that petri plate dish is there on petri dish, there was a fungus growing called as Penicillium notatum. Around the fungus, there was no bacteria. Then he started observing, why there is no bacteria around the fungus? Is fungus doing something to the bacteria? Is the fungus stopping the growth of bacteria? And that led to the discovery of antibiotics. And these antibiotics have saved so much of lives in your World War II. Okay, in World War II. Okay, so antibiotics, students, antibiotics. Listen to this. So this Penicillium notatum is your fungus here. It is growing here. Around the fungus, there is no bacteria only. No bacteria. Staphylococcus is not growing only. Why? Because this Penicillium notatum is actually killing the other organisms around it. Now, this is an example of amensalism actually. Amensalism. Amensalism is an interaction where one species is neither benefited nor affected, but other one is affected. Amensalism, students. Amensalism. Right? Amensalism. Example of amensalism. Commensalism, students. Commensalism is orchid going on a tree. Then we have the different types of your what is the students? Sea anemone as well as clownfish. Then we have your sea egret growing on a cow. All examples of commensalism. But this is the example of amensalism, students here. Students, is my voice cracking right now? All of you students in the chat? All of you, is my voice cracking, students? All of one second, let me just change the mic. Now, is it fine? All of you. I think it should be fine right now. I changed the mic, don't worry. When I change the mic, don't worry, students. Now, is it breaking right now? Fine? Proper? All of you? Fine, no, tell me yes or no. Tell no something. All of you students, this is what I don't. I want in my class to be energetic. Students, when I used to teach somewhere else, I want my students to be very active in my class. I will keep on asking them questions. They should always talk to me. I used to go sit next to them and talk to them. So I want all of you to be like that. Talk to me always, right? Now, this is your Penicillium notatum, students. Now, but the entire application part was done by your Ernest Chain and Howard Walter Florey. So Chain and Florey, along with the Alexander Fleming, all three of them won a Nobel Prize in your 1945. Nobel Prize winners, all three of them, Nobel Prize winner students. There are different types of antibiotics. You can see, students, different organisms are there. It is not given in NCERT. There is gentamicin, streptomycin. Students, these two are celebrities. All of you, these two are celebrities here. Okay, celebrities of your antibiotics. All of you might have heard about them. I have only taken streptomycin so many times. I have only taken gentamicin so many times. Right? These are celebrities here. The celebrities, students, celebrities here. Now, what is the use of antibiotics? Obviously, against the different types of diseases, bacterial diseases, plague, diphtheria, that is your gul ghotu. Then we have whooping cough, that is khali khasi, leprosy, that is kush rog. So against all of these are your antibiotics can fight against them. Very good antibiotic, students, very good. Okay. Now students, now. Antibiotics is done. It's done. Students, shall we start the last topic for today, students? Shall we start the last topic for today? Yes? Shall we start, students? Shall we start? After this, I have a surprise for all of you. After this, I have a surprise for all of you, students. Surprise for all of you. Surprise for all of you. Surprise. Students, shall we start? All of you. Students, shall we start the last topic called chemicals? How microbes are used to make certain chemicals also. Yes or no? Obviously, students, this is useful for board also. Both also you can study the same thing. Whatever notes I give, students, whatever notes I give, it can be used. Okay. Thank you, students. Now, chemicals. Now, we have three main categories here. Direct chemicals. Some of them are enzymes. Enzymes are basically your from your microbes. Bioactive molecules from microbes. From microbes only, students. Now, the first is the different types of organic acids from microbes. So, organic acids from the microbes. Here are the four different examples. All of you know lactic acid. Lactic acid is produced from, all of you in the chat who can tell me the answer? Lactic acid is from LAB, lactic acid bacteria. From lactic acid bacteria, we get your lactic acid. Then butyric acid from Clostridium butylicum. See, BUT. All of you can remember. Now, acetic acid by Acetobacter aceti. So, aceti, you can remember. Now, these three are easy to remember. This one you might find difficult. That is, citric acid is from your Aspergillus niger. This is different because it is a fungus, while Clostridium as well as Lactobacillus and Acetobacter, all three are from bacteria, students. All three are from bacteria, while citric acid is from a fungus, that is Aspergillus niger. Now, students, I'll give you exactly 10 seconds to look at the board and try to remember it right now itself. Time starts now. Okay, students. Students, I will give you DPPs in my Telegram. I'll give you DPPs in my Telegram itself. I have NEET PYQs today at the end of the class. I'll give you NEET PYQs also. Okay. So, 10 seconds, time starts now, go. All of you look at the images and try to understand, students. Try to remember the examples. Done? 10, 5, 4, 3, 2, 1. Students, tell me, from where do we get acetic acid? From where do we get acetic acid? Answer in the chat, all of you. Answer in the chat. From where do we get acetic acid? From where do we get acetic acid? In the chat. Now, students. Now, there are different enzymes. Lipase is an enzyme which can be used to clean detergents. It is used as a detergent, used to remove oil stains and everything. There is pectinase, proteases used as a clarifying agent. Then we have streptokinase. I'll teach you one by one now, students. I'll teach you one by one. Okay. Now, students, understand. First one, students. First one is called as lipase. Lipase can be used as a detergent. This lipase enzyme can be used, students. It can be used to remove the oil stains. Oil stains from the where? Oil stains from where? The dirty clothes. Oil stains from the dirty clothes, right? That is use a detergent. Then we have the pectinases. Pectinases and fruity. Students, have you ever drank fruity or Tropicana? All of you have drank Tropicana and fruity? No. This Tropicana and fruity, do they have any pulp inside? Or they're very, very smooth, they're very juicy, they do not have anything in the middle? Right? They don't have any type of your fiber inside. Yes or no? Now, if you make mango juice in the house is different, fruity is different. Why? Because, students, in fruity and all, they add pectinase and proteases. Enzymes which cleave pectin and peptide bonds respectively, clarifying the juice. So the fruity and all becomes very clear. That is clarified it becomes. Because of the pectinases and proteases, the juice becomes clear because of these enzymes. They are obtained from your microbes again. Okay. Now students, what about streptokinase? Streptokinase is obtained from Streptococcus. Please remember that. Now, what is the main function of streptokinase? Streptokinase is a clot buster. That is, if this is a blood vessel, if there is a blockage like this, blockage in a blood vessel, if you take a tablet of streptokinase, right, this tablet will completely break open the blood vessel and the blood flow can happen very easily. So this blockage can be removed by streptokinase. It is obtained from Streptococcus. So Streptococcus enzyme is the streptokinase clot buster. Okay. Now students, next one. Now students, let's say my phone is becoming very old now. My phone is becoming very old. Old phone I have. Okay, this is my phone. Those of you wondering, this is my phone right now. Now let's say I want to buy an iPhone now. So very costly iPhone. So what do? So obviously to buy iPhone, I will remove my kidney, I'll sell my kidney and I'll buy an iPhone. So I have given one kidney I got an iPhone now. Oh, I'm like, oh, iPhone I got now. Now I feel like one kidney is not enough. Now what do? When my sister is sleeping, I will take one kidney from her, obviously giving her anesthesia, and I will take her kidney and fix it to mine. Now, but what happens is that when I take my sister's kidney, no, I'll take her kidney and put it into mine. There's a problem. The problem is that my body will reject the kidney because it is not my own kidney. My kidney is sold already. So I have to suppress my immunity now. I have to suppress my immunity, that is to prevent graft rejection. To prevent graft rejection happens, right? Graft rejection happens. Now to make sure it doesn't happen, we'll suppress our immunity. To suppress our immunity, no, we take something called as cyclosporin A. Cyclosporin A is a immunosuppressive agent. To make sure I will get my sister's kidney to me, I will take one cyclosporin A, student. I'll take cyclosporin A. Okay, and this cyclosporin A is obtained from Trichoderma polysporum, which is a fungus. Cyclosporin A from Trichoderma polysporum, which is a fungus here. So to make sure our immunity is suppressed. Students, clear all of you? Clear? Thumbs up in the chat, all of you. Clear? Okay. Now students, with that chapter for today, class is over. Now, shall we start some PYQs? Students, for 10 to 15 minutes, let me do some PYQs, for 10 minutes PYQs. This much is today's class. Next class on Friday, I will finish the entire thing. On Saturday morning 11 a.m. I will take a class. Saturday morning 11 a.m. on top 30 questions from human reproduction. Okay? Yes. We will start now, student. We will start question now. Okay? Now, shall we start? Questions? So my voice breaking, students. I think there's something wrong with my mic today. Okay, students. I hope it doesn't break now. Okay? Shall we start? Okay. Now, here is the first question. Time starts now. Go. First question. Clostridium butylicum. I told you, students. Clostridium butylicum. Beauty. Yes or no? Okay. So A is going to be 3. If A is going to be 3, can I say this option is gone? This option is gone. Okay. Saccharomyces cerevisiae, brewer's yeast. Okay, brewer's yeast. Trichoderma polysporum, we just now learned cyclosporin A. Streptococcus will give you streptokinase. We learned here only, students. We learned it here only. Oh, students, we missed one more. We missed one more also. Bioactive molecule. That is your statins. Statins, students. Statin is obtained from where? Statin is obtained from Monacis purpurus. It's a fungus again. Monacis purpurus. Statins is a blood cholesterol lowering agent. Students, remember this also. Students. Statin is a blood cholesterol lowering agent. Statin from Monaskis purpurus. Then we have cyclosporin A, Trichoderma polysporum. We have streptokinase from Streptococcus clotbuster. Okay. Students, the answer is option number 2 here. Okay. So A is 3, B is 1, C is 4, D is 2. Okay, 2 is the answer, students. Okay. This question go. Is the same question? No, different question, student. Different question. Students, start, all of you, start. Saccharomyces cerevisiae, ethanol. Okay. Aspergillus niger, citric acid. Trichoderma polysporum is cyclosporin A. Clostridium botulicum is citric acid. Yes or no? So what is the answer now? A is going to be 3. If A is going to be 3, we have only 2 options there. Okay. And B is going to be Aspergillus niger. So BUT. D is going to be 2. So I got my answer. So what is the answer? So what is the answer? All of you tell me. B is going to be 2. B is going to be 3. A is going to be 3. B is going to be 1. C is going to be 4. That is your option number 1. Students, identify the microorganism which is responsible for production of immunosuppressive agent. Students, all these are NEET PYQs. All of these are NEET PYQs. All of you. Immunosuppressive agent. Cyclosporin A is got from your Trichoderma polysporum. Okay. Students, solve this question again. Students, in this chapter you get a lot of match the followings. Aspergillus niger from citric acid. Acetobacter aceti. Clostridium botulicum, that is your butyric acid. Lactobacillus, lactic acid. Yes or no, students? What is the answer now? Students, what is the answer? Obviously 3 here. So let me just check. Aspergillus niger, A is going to be 3. Yes, A is going to be 3. Only one option is there. A is going to be 3. Acetobacter aceti, C is going to be 1. Clostridium botulicum is going to be butyric acid. C is going to be 4, and D is going to be 2. So same thing only. 3 only. 3 is the answer. Okay, students. Solve this question, all of you. Which of the following commercial blood cholesterol lowering agent? I told you. It will decrease the cholesterol in the blood. It will decrease the cholesterol in the blood. Blood cholesterol lowering agent. Which of the following is a blood cholesterol lowering agent? So quick. Blood cholesterol lowering agent. That is your statins. Very good. Next question. For commercial and industrial production of citric acid, which of the following microbe is yours, students? Citric acid, a fungus. We get from, students? We get from Aspergillus niger. Very good, students. Cyclosporin, students. Again, students, do you see a pattern here? Students, do all of you see a pattern? Every single year NEET examination, same pattern is followed for these type of questions. These type of questions, students. Cyclosporin, all of you know. Trichoderma polysporum. Okay, students. Solve this question, all of you, students. Solve this question. Take your time and solve this question, students. Take your time and solve this question. See, Saccharomyces cerevisiae is used for ethanol making. Okay. Monaskis purpurus, I told you just, student. Monaskis purpurus. Statins. We learned here only. Monaskis purpurus we learned here only. We learned here only. Okay. Trichoderma polysporum, immunosuppressant, cyclosporin A, immunosuppressant. Propionibacter shermani, that is your cheese. Cheese. Students, think again, try answering properly. See, A is going to be 3, right? So, this option is gone, this option is gone. Okay. Monaskis purpurus, statins, blood cholesterol lowering agent. Trichoderma polysporum, immunosuppressive agent. Immunosuppressive agent. Right. Propionate Sharmani, ripening of cheese. DS2. So what is the answer? What is the answer, students? What is the answer? Option number 1. Okay, students. So learn the example, that is very important. Monaskis purpurus, statins, cyclosporine, trichoderma polysporum, streptococcus, same thing. So with that, today's class is over, students. Before I go, I have a surprise for all of you. Students. Give me 1 minute. I have a small surprise for all of you, students. Small surprise. I'll be giving you all of this in the end of the class, at the end of the chapter. I'll just show it to you right now, but you'll be getting it at the end of the chapter. Okay. 1 second, students. Students, see this? All of you? Students, it's visible to all of you? Students, it's visible to all of you? Students, tell me in the chat quickly. Tell me in the chat, is it visible? Let me try decreasing the brightness here. Okay. I've decreased the brightness a lot. So try see, students. See this. Okay. Students, what did we learn today? Okay, I'm going to go black here. I hope it is visible to all of you. Okay. I hope it is visible to all of you now. You won't be able to see me, but this is the more important. Okay. See, students, here we learned today's class, we learned microbes in household product. Yes or no? Microbes in household product. Curd, bread, toddy, Swiss cheese. Curd, lactobacillus. Bread is your Saccharomyces cerevisiae. Toddy, we don't know. Generally, it is a type of yeast itself. Students, it's not clear to all of you. Let me just zoom in and show you, students. Let me just zoom in today. Later on, we can just see later on. 1 second. Now it might be visible, students. Okay, we learned this today. Microbes in household products here. Swiss cheese, Propionibacterium shermani. Propionibacterium shermani is Swiss cheese here. Okay. Then, students, today we learned this also. Microbes in your industrial application. In industrial, we have what? Fermented breweries is your this Saccharomyces cerevisiae. That is again basically your yeast. Then we have antibiotic, Penicillium notatum, organic acid, Aspergillus niger, fungus, citric acid, Astrobacter aceti, bacterium, acetic acid, Clostridium botulicum, bacterium, butyric acid, Lactobacillus, lactic acid, enzymes, lipase, pectinase, clarifying bottle juice, streptokinase, streptococcus used as a clot buster. Then bioactive molecules, statins, Monaskis purpurus, blood cholesterol lowering agent, cyclosporine, Trichoderma polysporum, immunosuppressive agent, students. So, do you realize, students, the entire chapter I'll be giving you it as the entire chapter, students, I'll be giving you as a mind map. Students, the entire chapter I'll be giving you as a mind map at the end of this chapter. Okay. So students, thank you so much, students, for today's class. Again, students, it's a reminder for all of you, right? If you are a class 12 student and if you are looking for a proper structured study, if you want to finish your entire syllabus by December itself, trust me, DNA batch is worth it. I've been teaching anatomy there. I've been teaching anatomy for certain from past 3 classes now. I'm going as detailed as possible, and I've spent around 1.5 hour, 1.5 hour, 1.5 hour, right? 4.5 hours, 4.5 hours to teach 3 pages of NCERT. That detailed and patient I'll be going in DNA batch. Students, if you're not a part of DNA batch, please start, please join. I'm telling you, it's going to be worth it, right? How many parts? How many? Next part I'll finish the chapter. That is day after tomorrow. I will finish the chapter day after tomorrow. Students, let me know in the comment section how was the class. Now here is a homework question, students. Homework question to all of you. Students, the homework question is that the homework question in the comment section, you tell me, students, tell me, right? Tell me all of you in the comment section, right? Which acid is produced by lactic acid bacteria? Very simple question. Which acid is produced by lactic acid bacteria in the comment section? And because of that acid, what happens in milk? Because of that acid, what happens in milk? Okay, students. So let me know in the comment section how was the class. Your homework question also is given to all of you, students. Right? So thank you so much, students. Thank you so much. I will see you in the next class. Until then, keep studying. Do not waste any time. Okay. So students, bye bye all of you and take care. Bye bye, students. Bye bye all of you.